hello, welcome to part 4 of AQA Biology Paper 1 2018 series. Okay, number 5. Plants can be infected by fungi, viruses and insects. Aphids are small insects that carry pathogens. There's little green insects that ladybirds like to eat. Figure 6 shows an aphid feeding from a plant stem. Sharp mouthpiece, plant stem, aphid. An aphid feeds by inserting its sharp mouthpiece into the stem of a plant. Give the reason why the mouthpiece of an aphid contains a high concentration of dissolved sugars after feeding. So what it is it's piercing the phloem. So the mouthpiece has pierced, or it's entered, or it's fed off, pierced the phloem. So that's where all sugars are transported in the plant, okay, the phloem. Plants infected with aphids may show symptoms of magnesium deficiency. Magnesium deficiency symptoms include yellow leaves and stunted growth. Explain how deficiency of magnesium can cause these symptoms. Make a bit of room here. Okay, so let's have a think about the yellow leaves. So yellow leaves due to lack of chlorophyll. Okay. So less light absorbed, light energy. So lower rate of photosynthesis. Okay, lower rate of photosynthesis, which means so plant makes fewer sugars for conversion to proteins, which leads to stunted growth as protein is needed for growth. Plants and animals really, but protein is needed for growth. Oh, needed, D on the end. Okay. A farmer thinks a potato crop is infected with potato virus. Why? PVY. The potato obtains a monoclonal antibody test kit for PVY. To make the monoclonal antibodies, a scientist first isolates the PVY protein from the virus. Describe how the scientist would use the PVY protein to produce the PVY monoclonal antibody. Okay, this is quite an unusual one. Okay, so it's kind of like you just have to know this process. So inject the protein into a often used for laboratory stuff, a mouse. So combine lymphocytes with cancer cells to make hybridoma cells. So find a hybridoma in a little space. Little space. Right, find a hybridoma which makes a monoclonal antibody specific to PVY. Then the scientists close. Hang on, just more space again. It's quite a few marks, 4 marks as well, isn't it? So the scientist clones the hybridoma to produce many cells to make the antibody.